speakers who have come here in space, and they are addressing us on this Cancer Day. I am not a cancer biologist. However, I look at cancer as an aberration of genomes. And therefore, in this very short presentation, I'm not going to take you through any data. I'm only going to take you through some ideas coming from a genome biologist. And when a genome biologist looks at cancer, it comes out as a marvelous design of Darwinian evolution. And that is what I want to just share with you in this presentation. These are only few slides I have. And in these few slides, I want to essentially give you one or two very important take-homes. So here we go. I go to my second slide. Has the slide moved?
Yes, sir.
Yeah. Okay. So in this biology 101 slide, what am I showing? I'm showing that genomes are quite dynamic. They accumulate mutations, and these mutations become genome aberrations. This is part of normal lifestyle of genome homeostasis. As we speak, our genomes are changing. However, many of these mutations do not lead to any serious phenotypic consequences because things are taken care of by very elegant mechanisms. So this is the basal level of genome changes that keep happening all the time in the cells. These changes also are essential drivers for cellular plasticity. In some sense, during differentiation and de-differentiation, etc., changes via epigenetic and transcriptional landscape changes. So this is the basal biology that keeps happening in all the normal tissues. So normal tissues in some sense are not static in biology, but very, very dynamic at the genome level. Things are changing. However, when these cells are changing intrinsically, dynamically, and are under homeostasis, they also come under the influence of environment. They can be micro-environmental changes or mega changes. When these cells are subjected to environmental effects, many possibilities arise. There can be metabolic changes, there can be genomic changes, there can be epigenetic changes, etc., etc. So ultimately, cellular changes are intrinsic as well as extrinsically driven. So that is the basic biology 101 of normal cells. However, these normal cells kept under check can go haywire under special conditions such as aging, high mutational loads, under some special tissue environments, etc., etc. And then you have what are called excessive growth in the tissues. Excessive growth in the tissue is a disease in human-centric context. However, in a Darwinian context, it's a winner because it's a growth. It's a growth that is using nutrients that are available. So it's a winner in Darwinian world. And these growths can be quite complex many times. They are linear growths. Tumor can be quite homogeneous, reasonably. The changes inside the tumor tissue can be somewhat heterogeneous. You can give rise to clonal variation within the same tumor. And this branched evolution can be quite complex sometimes. It can be very simple some other times where most of the tissue is clonally okay, but for some variations here and there. And then some of these changes can even be very neutral without giving rise to any phenotypic consequences. In fact, some of our normal tissues have a lot of genomic changes, and each tissue is a collection of clonal populations. So you can see that maybe lots of neutral changes giving rise to lots of cellular changes without any phenotypic consequence. Everybody is normal. So you can see that changes can happen giving rise to phenotypic changes or no phenotypic consequences as well. So this is part of life, and this particular part of life can go haywire in certain contexts. Let us look at those contexts. Before we look at those contexts, let's look at what is evolution. Basic design of evolution is captured in this slide. You have genes as units. The only function of genes is to express and multiply multiple times. Gene units have to replicate and replicate and replicate in tissues and organisms. When these genes replicate, copy number obviously goes up, but gene functions gives rise to phenotypes, which is what interacts with the environment. So genes interact with the environment through phenotypes. And environment impacts the genes via phenotypic selections. You can see here. These phenotypic selections are the one that impact environmental changes which are induced on the genes. So you have genes getting multiplied, phenotypically cross-talking with environment impacting phenotype, giving us to selections. This whole cycle is the primary cycle in evolution, going through multiple times. Okay, so this is the basic design of evolution. So multiplication and selection happen successively. Now, in this backdrop, let us look at a situation. Cells have to go through proper cellular replication regimes, which is what is called cell cycle. And in every cell cycle, there are checkpoints to take care of intrinsic aberrations that can happen. Multiple checkpoints are there. Let us look at this normal white car, which is a healthy car. This car, when it undergoes some genomic aberrations, it goes through a checkpoint arrest. That is, in the car that is running on the highway comes out of the exit, gets repaired in the checkpoint, and comes back and goes onto the highway, goes on cycling. A normal car has a time window to go through checkpoint arrest where it is repaired. This is the normal car, normal healthy tissue. However, cells which are somewhat aberrant, which cannot repair themselves, the black car cannot go through checkpoint because it is repair compromised. So it is halted in normal environment, whereas the normal cell keeps on recycling because it has this luxury of going through a checkpoint repair. Now imagine a situation where the same normal cell is under the onslaught of mutations or aging, etc., etc. So lots of mutational load happens. Then the consequence is the normal car gets stuck in the checkpoint repair. It's all the time under repair because constant damages keep this car, normal car, under checkpoint arrest. It is in the garage all the time. However, the black car, which was repair compromised, gains advantage. In this high mutation load, which is what is the cancer progression, the car which is repair compromised gains fitness advantage through which it starts cycling. When the normal car is stuck, the mutationally loaded aberrant mutational cell, aberrant repair cell gains fitness advantage, which starts recycling and becomes excessively grown. This is the tumorous cell. So when does the tumorous cell arise? Tumorous cell generally arises when there is a compromised repair. Now the same thing is modeled in the bottom portion of the slide. Am I visible, audible? Everything is okay to my audience?
Yes, sir.
Okay.
Yes, sir.
Now look at this dotted line. Dotted line is a classical reciprocal relationship of a normal cell. As the error rate increases from left to right, the replication rate plummets more or less linearly. You can see the dotted line. This is the normal healthy cell showing classical reciprocal relationship. As the mutation load increases, replication rate decreases, which is what is the normal cell getting stuck in the garage. Look at this non-linear dark curve. This is the curve represented by a repair compromised cell. What is the consequence? The repair compromised cell shows rapid decline in replication in the early stages of mutations. As the mutational load increases, it plateaus and it takes off here. Now this graph, which is repair compromised model, gets lifted up as the repair becomes more and more severe. You can see here, this black curve is nicely sitting at the bottom. This black curve lifts up itself as the repair becomes less and less efficient. It becomes more and more compromised. As the cells become more and more cancerous, this black curve lifts itself up, comes to a stage where it becomes much more aggressive than even the dark dotted line. So much so that this black graph continuous graph lifts itself much above the normal dotted line. This is the consequence of cancerous Darwinism, where heavily repair compromised cell gains tremendous advantage of replication, excessive against lots of mutation load. That Darwin has woken up in full gusto in these repair compromised cancerous cells. So the selections give enormous fitness advantage to this black car, which is highly repair compromised. In spite of the mutation load, it keeps cycling. It keeps cycling in the cell cycle, gives rise to a lot of multiplication. So this is a Darwinian selection that gives rise to excessive replication of cells, which is heavily repair compromised under mutation load or under heavy aging load. Consequently, these cells have such high fitness advantage that they keep on replicating. Nothing can stop them. However, classical healthy cell has stopped replicating and it is already touching the bottom of the replication level. So you can see that this is an interesting change in the function of the repair compromised cells. Low repair to moderately low repair to very highly compromised repair. So essentially, you have cancerous cells which are highly repair compromised in multiple ways. Mechanisms are too many to depict. Multiple ways the repair compromisation happens, and then the cells essentially go berserk because Darwinian selection gives them enormous fitness advantage. The normal cell is just simply resting there. This is an important take home for all cancerous tissues where Darwinian selection brings in so much of fitness advantage to these heavily compromised cancer cells. Consequently, what happens? The initial chromosomal errors which happen in the precancerous cells give rise to loss of function, and then you have this kind of compromised replication. However, these chromosomal errors become very aberrant chromosomal errors, which give rise to selection, so that this checkpoint protein, MCP is a checkpoint protein, mitotic checkpoint protein, becomes mutated. So this actually is the culprit why the repair causing proteins become compromised so heavily that a compromised replication now becomes a robust replication in the cancer tissue. So this is the bottom portion of the model where aberrant chromosomal errors give rise to tremendous driver mutations as well as passenger mutations that gives rise to heavy rounds of replication, which was otherwise compromised in normal cell or feebly replicating cells. So you can see a demon has woken up by Darwinian selection, where multiple mutations give rise to total loss of repair functions, and the system is more or less has gone amok. It's just one example of mitotic checkpoint protein. There are many examples of this type. Consequently, even in aging, this model applies. Repair activity is significantly suppressed with increasing age. There is heavy data on this. All of us, when we get aged, our repair efficiencies go down. Consequently, malignancies come up much later in age, because when the repair efficiencies go down, this model applies. And when that model applies, you have lots of these tumor cells coming up. And when the tumor cells come up, this model applies where chromosomal errors become more and more aberrant. The repair genes themselves get mutated, giving rise to tremendous heterogeneity in the tumor cells. Some of them get selected and they become heavily aggressive. So there is a fair amount of heterogeneity in the tissue environment, within the tissue context. There are serious winners, which, depending upon the micro environment of the system, some cells are selected and they become aggressively growing. So there is a role of intrinsic changes at the genome level and extrinsic changes which are impacted by the microenvironment. This is the crucial crosstalk that happens in metastasis. So this captures lots of interesting changes that people have shown. over several decades now. Clonal evolution of tumor cell by successive mutations. Successive mutations accumulate by a combination of mutations. Some of these cells go over the selection barriers. Selection barriers can be of multiple types. Metabolic barriers can be there. Other tissue barriers can be there. Some cells go over that barrier and come out and aggressively propagate. Sometimes therapeutic chemicals can impose barriers. And when therapeutic chemicals, chemotherapeutic chemicals impose barriers, selections give rise to havoc in genomic changes. These are what are called heavy chromosomal rearrangements. In fact, whole genome doubling can happen. You can see here, whole genome gets doubled. So the chemotherapeutic agents are really double-edged swords. Chemotherapeutic agents, of course, halt these cellular proliferations, but also impose selections for very aggressive genomic changes such as these. And these aggressive genomic changes give rise to drug resistance mechanisms. And these are very well described now, and multiple examples are coming out in the context. So much so that as a function of drug treatment, clonal propagation happens, and you have heavily drug-resistant clones developing at the end of the selections, Darwinian selections. In the very beginning, perhaps there are drug-tolerant populations which are useful to target. But at the very end, when Darwinian selection goes through serious level of maturation, then you have very heavily drug-resistant clonal populations, which are pretty much refractory to most treatments. This is a real challenge in cancer biology. How do we target the early population which are more or less drug tolerant, where the genome changes are somewhat less severe and more amenable for drug treatments? Whereas when the selections become very heavy and these genomic changes are so abnormal that any treatment doesn't really target them and becomes excessively drug resistant. So one of the challenges in tumor biology is to understand the tumor heterogeneity causing heavy tumor resistance. And before this heavy tumor resistance happens, can we understand the genomic basis of drug tolerance causing mutations which we can target successively? This is the Achilles heels in cancer biology. If we can do that by multiple mechanisms, then probably we can tame this particular selection process of early Darwinism. So what is the take home of whatever I said? Is Darwin useful for us to understand biology, cancer biology? Certainly yes. Is Darwin useful in understanding some level of therapy modalities? Probably yes. And that is what is depicted here. This is a little bit of a speculative slide now, which is getting backed up by a lot of data. You can see that conventional therapies give rise to treatment initiation, gives rise to mutant cells and minimal residual disease system here, MRD. And then if you impose drug selection, the chemotherapeutic selection, resistance comes about. And this resistance is a combination of multiple types of clonal changes, and you have a relapse with heavy clonal changes. This is pretty much irreversible at the genomic level, at tissue level as well. If you can bring about adoptive therapy, where your drug treatment gives rise to milder clonal system, which relapses, but then you continue the low doses of the treatment, it relapses, you continue low dose, it shows response, it relapses, it shows response, it relapses. So this is something that one has to understand at chemotherapy level. If we can adjust the dose at an adoptive therapy level, you can see the response, it will relapse. Again, if you keep up the pressure, not too high pressure, not too low pressure, you can keep up the response, relapse. This can continue for some time. This actually does give time advantage to the patient. Another approach is, once you have heavy resistance with minimal residual cells here, it of course acquires resistance in the due process. But if you hit with the second line of therapy, this tumor can regress. These are, of course, very well known, ABC, but these D and E modalities are coming up a little bit more recently these days. We don't fully understand this. These approaches are those approaches where we hit drug tolerant population. And in this drug tolerant population, second line of therapy, as well as certain monotherapy also gives rise to complete elimination of the tumors. This is where I think there is enormous window of opportunity to understand where and how we can apply drug therapies targeting selectively, tolerant cells rather than resistant cells. There is a time window which we need to capture. If we can enlarge that time window, we have good opportunity for treatment. So my take home is essentially this, where we need to keep mechanistic understanding of how this fitness advantage is gained by compromised pre-tumorigenic or tumorigenic cells, and this essentially gains so much of fitness advantage that replication goes through the window much above the normal cell, and that's how the excessive growth happens. That is number one take home. Number two take home is in this business of progression of drug resistant cells, drug tolerant cells, are there important windows for us to hit the system successfully, which is depicted in this model. So these are the two take homes that I'm really excited about, to think in terms of mechanistic details and see if we can contribute in terms of ideas. This is where I stop. I thank all of you for giving me this very small time window of talking to you and sharing some of these ideas. Thank you very much. I'm a little bit under time pressure, so I will not stay for too long. I'll sign off and I have to join some other meeting. Thank you very much. Namaste.
Thank you so much, sir, for joining us.
Thank you, sir. And
stop sharing. Yeah,
sure. Now, I would like to request my colleague from Department of Biochemistry, Professor Brahmanand Manavati, to introduce the next speaker.
Am I audible?
Yes.
Very much.
Yeah, okay. Thank you. So very good morning to all of you. And I take pride to introduce the second speaker of the occasion today, Professor Kumarvel Somasundaram. So before I introduce the speaker, let me say a few words about the significance of the day. So the cancer is deemed as the emperor of all melodies. Unlike the COVID-19 outbreak, which invoked catastrophic effect in the last 2 years, it is still on. Arguably, cancer is pervasive and entrenched in our society. So this vicious ailment is known to the humanity for nearly 250 years. Yet it continues to be unabated and haunts us. We are all aware that millions of people around the world succumb to this pernicious malaise. So the World Cancer Day is celebrated on February 4th every year since its inception in 2000, and it aims to save millions of preventable disease deaths every year by raising awareness and education about cancer. And not only that, and also pressing the governments and individuals across the world to take action against the disease. So on this special occasion, so we have just had tasted a few interesting thoughts on cancer by Professor B.J. Rao, our beloved Vice Chancellor, on the everlasting concept of the cancer as a Darwinian evolutionary model. And now we will have another reverberating and exciting talk by Professor Somasundaram. He is very familiar to the School of Life Sciences. And so let me give a brief introduction about him. So Professor Kumarvel Somasundaram is currently heading the Department of Microbiology and Cell Biology, Indian Institute of Science, Bangalore. So he obtained his veterinary medicine degree in 1985 from Madras Veterinary College, and subsequently he obtained his master's degree from master's degree in biotechnology in 1987, and PhD in bacterial genetics from Madurai Kamaraj University, Madurai. So after graduation, he obtained a postdoctoral training at Northwestern University and the University of Pennsylvania in cancer genetics before moving to the Indian Institute of Science in 1999. So the primary focus of his laboratory is the genetics of glioma. So one of the most common and inspirational and one of the primary adult brain cancers. So he made outstanding contributions to the field and is an exemplary and inspirational to many youngsters. So his unsatiated hunger for a better understanding of the glioma continues at ISC. So with this brief introduction, I warmly welcome you, sir, and thank you for joining. And now you may deliver the talk.
Thank you. Thank you, Brahmanand, for the nice introduction. And also, I would like to thank Dr. Bindu Madhav and Professor B.J. Rao, Vice Chancellor, University of Hyderabad, for inviting me for this meeting. Let me share my slides first. Screen. Is my slides are visible in a starter mode?
Not yet, sir.
Not yet. Okay, sorry.
You have to share a screen first.
Share the screen, sir. Yeah.
Yes. Okay. Are the slides visible in a starter mode?
Yes, sir. Yeah. We could see.
And the first slide?
Yes.
And in show mode?
No, I think you need to put it in the still.
Yes. And are the slides moving?
Yes.
Okay. Am I audible well?
Yes, sir. Yes.
Okay. Thank you.
Yeah.
Thanks again for inviting me for this meeting. The title of my talk, as you might have seen in the announcement, Cancer Stem Cells and Plasticity, who are the real culprits? The reason I put this title is while we have been succeeding in getting better cures, better ways of treating cancers, largely cancer treatment essentially results in reducing the growth of the cancer rather than completely removing the cancer. Okay, so essentially you are increasing the median survival, but you are not able to completely cure any cancer for that matter as of today. Okay, so that means there has to be something in the cancer which is responsible for this recurrence of the cancer. Okay, so cancer stem cells appear to be the right choice, but then there is this phenomena called plasticity, where one type of cell can become another type of cell. So with that introduction, I would like you to present my slides. Basically, I'm hoping to give you a different way of looking at the cancer than what everybody else looks at it. I hope I will succeed in that. Okay. So if you look at the biology of cancer, what you see on the left side is actually a pseudo-colored image of a glioma, the brain tumor. And the simplest way one can actually define cancer is an uncontrolled cell proliferation. Or you can also say, as Vice Chancellor said, it is actually a genetic disease because there are genetic changes which are occurring in the cancer cells compared to the normal cells. Okay, so if you look at a little bit more in better way of defining cancer, I've taken these words from this Peter Novel's paper published in 1974. Cancers arise from a series of mutations or genomic alterations that provide a cell with an extensive ability to evade pro-apoptotic and growth inhibitory signals and to be self-sufficient in growth signals that enable them to divide endlessly. Subsequently, a few more additional points were added to this definition by Bert Wogelson's group and subsequently by Robert Weinberg's group. And that is, additional genetic alterations also occur in these cells to promote other characteristics like enhanced angiogenesis, tissue invasion, and metastasis. These two groups have come up with models. Bert Vogelstein Professor Bert Vogelstein came up with this multi-step carcinogenesis model in 1990, wherein the normal cells, one mutation occurs which converts them from normal to hypoproliferative cell, and then subsequently additional mutations occur and finally resulting in completely metastatic tumor. So basically accumulation of mutations one after other results in the completely transformed cell with the metastatic capacity. And then Professor Robert Weinberg came in 2000 and then subsequently 2011. Initially they proposed there are 6 hallmarks. Later they have increased to 10 hallmarks. These are 10 different characteristics by which cancer cells differ from the normal cells. Okay, with this introduction, if you actually look at the cancer as a bundle of cells, okay, and how do they actually look like? Little bit go into details. If you suppose look at one first time ever a cancer cell is formed because of a genetic alteration, perhaps one single mutation, resulted in formation of the transformed cell. The cell starts dividing. As it divides, more and more genetic alterations occurs. There is a clonal evolution, and then there will be a subpopulation of cells with varying set of mutations that results in so-called genetic heterogeneity within a tumor. And that's one of the reasons people always thought why our treatments fail. People, treatment doesn't give a durable response. Always tumor recurrence occurs. Okay. One of the other reasons people thought in the last 2 decades why the cancer recur is the cancer stems hypothesis. Based on this cancer stems hypothesis, people have shown experimentally, there are evidences existing in a tumor. There are a small population of cells which occupy a small percentage of the whole tumor, which are called cancer stem cells. These cells alone are capable of initiating and maintaining the tumor rather than the remaining bulk of the tumor. How does it work? For example, if you have a normal stem cell, and then it gets mutated when it is exposed to carcinogen, and then results in the formation of the cancer stem cell. The cancer stem cell divides. As it divides, it also differentiates. I thought it's going to happen. So there is some problem in my animation. Okay, let me see whether I can go back. Okay, sorry. Essentially, the cancer stem cells, when they divide, keep dividing as well as they start differentiating to form so-called bulk cells. Okay, the bulk cells are not cancer stem cells. They are supposed to be the differentiated cells. They don't divide anymore. They are there to just form the bulk of the cell. Cancer stem cells, which are in small proportion, are the only one which are important in the cancer, and they are the one who can initiate the tumor and also responsible for maintaining the whole tumor. Unfortunately, the animation is not coming, so I will let me try again. Okay, so as they differentiate, form a lot of differentiated cells, the green cells, which form the bulk of the cells. So it is important to note, in any tumor, these cancer stem cells, which are red in color, they form a small proportion, yet they are the one which are very important. They alone can initiate a tumor, and they are alone responsible for maintaining the tumor, while the differentiated cells, which forms the bulk of the tumor, even though they form the bulk of the tumor, they don't seem to be that important according to the cancer stem cell hypothesis. Now, cancer stem cells are, why they are very important. One, of course, as I said, they alone can initiate a tumor. They are also called, in addition to cancer stem cells, they're also called tumor initiating cells, tumor provocating cells, different terminologies used by different scientists. And they are also, why they are, in other words, there are also other reasons why they are important. The current treatments actually do not kill the stem cells and they remain alive. They are responsible for recurrence. So all over the world, people are trying to find out drugs which can specifically target the stem cells so that you can combine with the current treatments to get the durable response. So although those kind of efforts have largely failed so far, still people are trying to identify drugs which can target cancer stem cells all over the world. So now, if you look at the tumor a little bit more deeply, here is a cross section of a brain, mouse brain, where the glioma is growing here. You can see the highly infiltrative cells. A lot of tumor cells are there. But you actually look at a little bit more closely, the tumor is just not made up of tumor cells alone. Complicating to that, if you actually look at a schematic view of a tumor, and in addition to the tumor which is there in the middle, which has the cancer stem cells as well as differentiated bulk cell, there are also stromal cells, for example, immune cells, and then the blood vessels, which are made up of endothelial cells, pericytes, and there are also normal cells in the case of glioma, normal astrocytes. So there are a variety of stromal cells which are also part of the tumor. So that results in the so-called tumor cellular hierarchy and heterogeneity. This is very important for me to explain the process of plasticity. So next slide, if you just see tumor cellular hierarchy and heterogeneity, how does it come? So if you look at the hierarchy, I told you the previous slide, there are cancer stem cells, differentiated cells, stromal cells. Which of these cells are very important? Which is on the top of the hierarchy? If there are like so many different types of cells in the tumor, which is on the top, which is the most important cell in a tumor? Okay, it is always cancer stem cells which appear to be on the top of the hierarchy, because if you divide these cells to different groups, you take a tumor, whole tumor, divide them into different type of cells and check whether which type of cell can actually initiate a tumor in animal model. It is always a cancer stem cell which alone can initiate a tumor. That's why it is on the top of the hierarchy. That's fair enough. Number two, the plasticity brings a different view of looking at it. So the meaning of plasticity is that cancer stem cells, for example, they can actually differentiate to form the non-cancer stem cells. That's how, of course, the tumor is formed. And it's also known recently, both stem cells as well as the non-cancer stem cells can actually differentiate to different lineage, like endothelial cells, and also different type of other cells also have been demonstrated, while the conversion to endothelial cells is very well documented. So this brings in the so-called heterogeneity. Okay, so it is very difficult to really assess in any given tumor what proportion the stem cells versus non-stem cells and the trans-differentiated endothelial cells, leave alone other stromal cells which are coming from the host. So because of this, the title I put it, cancer stem cells and the associated plasticity, which makes this tumor highly heterogenic, and who are the real culprits. So I would like to present some data generated from my lab to give a different view of looking at a tumor. Okay, for example, if you have a normal stem cell which undergoes mutation to create a cancer stem cell which divides, and then also differentiates to form the tumor. I showed you a few slides ago. Now, if you look at in a tumor, bulk of the tumor is made up of the differentiated cells. What is the function of these bulk cells? Are they there to just form a mass, or do they have a function? This is a question we asked. Of course, we agree cancer cells are the most important one, and they are on the top of the hierarchy. They alone initiate a tumor, because people have shown if you actually inject non-cancer stem cells or the differentiated cells or so-called bulk cells, they cannot initiate a tumor. I agree, totally agree with that. But then the bulk cells occupy the most of the tumor. And is the function is to just simply form a bulk of the tumor, or do they have any function? This is a question we asked in our study. Okay, so in our lab, we work on glioma cancer stem cells, because you work on glioma. And okay, just not moving. Okay, so we work on glioblastoma, which is a grade 4 glioma. Glioma is, of course, the tumor of glial cells in the brain. And I showed you the incidence, and it is one of the most aggressive cancers. Current treatment is surgery, radiation, chemotherapy, like in any other cancer. And the survival is very, very terrible. 50% of them will die within 15 months. Remaining 30 to 40 will die within 2 to 3 years, and the other 10% will die within 3 to 5 years. Essentially, by 5 years, everybody will die. And the reason being, one of the most important reason being, all GBMs, glioblastoma, actually recur. There are many reasons why GBMs recurs. Obviously, one of the important reasons is presence of cancer stem cells, the glioma cancer stem cells, which are appearing to be important for the GBM recurrence, because they resist the traditional treatment and results in the recurrence. So the first report of glioma stem-like cells, or the glioma stem cells, happened in 2004. 2 different independent reports. They essentially showed CD133 membrane marker positive cells, which can grow as a neurosphere, actually constitute the cancer stem cells of glioma. Although there are some conflicting reports came afterwards, let us take it for our purpose of discussion. CD133 positive cells, which can form neurospheres, are the ones which form cancer stem cells of glioma. And then 10 years later, this landmark paper came, where they very nicely showed glioma stem cells can be grown as neurospheres, and these neurospheres can be made to undergo differentiation in vitro to form so-called differentiated glioma cells, which grows as a monolayer in fetal cough serum containing media, and then subsequently they can undergo dedifferentiation to form induced stem cells, and for which they identified 4 transcription factors, something similar to Yamanaka factors. These are called glioma reprogramming factors. Interestingly, when they compared the epigenetic profile of the original stem cells compared to the induced stem cells, the epigenetic profile looks similar and almost similar to each other, suggesting the process of differentiation and dedifferentiation is completely reversible, and that is because the process involves epigenetic change. that's good because it's easy to target a epigenetic change. So now in my lab, actually we get freshly resected glioma samples and we try to grow them as stem cells in the form of neurospheres. We also allow them to form monolayers through differentiation, and we call them as the glioma stem cells, GSCs, and differentiated glioma cells, DGCs. And these two terminologies I will be using very often to describe glioma cancer stem cells as a GSC and glioma bulk cells, differentiated glioma cells as a DGCs. GSCs versus DGCs. We have a bunch of GSCs and the derived GGCs from the GSCs, and we try to compare them for our studies. So what we did is we took a bunch of glioma stem cells and the differentiated glioma cells, and we tried to look at the secreted proteins particularly. Reason why we wanted to look at secreted protein, let's say we inject a bunch of stem cells into the brain of a mouse, initially for the brain, the cells to form a tumor, to make a bigger tumor like this, it's important they get angiogenesis to get the nutrients. So it is important to get the angiogenesis activated, and that's probably can be done by secreted proteins much easily than membrane or intracellular protein, and that's the reason why we looked at secreted proteins. So we did a label-free quantitative proteomics, and then we identified by comparing secretome of 3 different GSCs and their corresponding DGCs, so many number of proteins with the 2 unique peptides, and 331 peptide proteins appear to be differentially present between the conditioned media of GSC versus DGC, and that is shown here. These are proteins which are highly abundant in the secretome of GSCs, while these are highly abundant in the DGCs. So while there are many proteins from this category we are working on, because we wanted to study primarily the role of stem cells, glioma stem cells in the biology of glioma, we got interested at one particular protein called fibromodulin. And I'll tell you the reason why we took up this protein, which is present in high abundance in the conditioned media of differentiated glioma cells. Okay, what is FMOD? FMOD is actually fibromodulin, collagen binding leucine-rich proteoglycan. I'm sorry, I think I pressed this one. I have to do this sharing again. Sorry.
Yeah. No problem.
Okay. It's not allowing me to share. Okay. The share function is not working.
Try again, sir, once again. Yeah.
I know, I just tried.
Yeah.
So only the cancel button is actually, okay, I'll do the canceling and then do it again.
Yeah,
Yeah,
maybe.
maybe.
Yes. No, share button is not getting lighted.
Exit and come back.
Exit and come back, sir. I think, yeah, that's
I know, I tried that.
Okay.
No, just, okay, okay, fine.
Yeah.
Can you see the slides?
Yeah, I think it's getting ready, sir. Yeah, sir. Yeah.
Okay, sorry about the disturbance.
Put the presentation mode, sir. Yeah.
Yes. So it's a small leucine-rich repeat proteoglycan, and it is present in the extracellular matrix. Okay, so we have earlier published a paper on this protein. We basically found that this particular protein expressed in high amounts in glioblastoma grade 4 glioma. And what we show in that paper is the expression of this protein is under control of TGF-beta, and it activates integrin, FAC, RO-RAC signaling to activate glioma cell migration. Interestingly, we found the protein has no effect whatsoever on the proliferation. Does not affect the proliferation. It only induces the migration. Okay, so we want to ask a question: a protein which is expressed only by the stem cells, the differentiated cell, does it have any role in glioma growth, tumor growth? So before we answer the question, we just validated. We can see that the transcript levels are higher in the differentiated cells. Protein level is also higher in the intracellular as well as secreted proteins in the DGCs. And we then ask the question role in the glioma, and you can see E251 cells, which expresses more amounts of FMOD. When you silence them, yes.
I think cells are, I mean, slides are not moving, sir.
Okay. How is it now?
No.
No?
No. It got frozen actually.
Okay. So I have to.
Yeah, these are technical glitches are very quite common.
I know. Is it moving now?
No, sir. I think you need to put again.
Okay, sure. So, okay, I will do it again. Okay, is it visible?
Yes, sir. Yeah, I think we put it in the presentation mode. Yeah.
Okay, sure.
Right.
So basically, just to quickly summarize this slide. So when we silenced this protein, we found out that silencing leads to the complete loss of the tumor growth. That was a surprise. That was a surprise for us because here is a protein which is secreted by differentiated cells, not by the stem cells, which does not affect proliferation. It only promotes cell migration. Why silencing of protein of such type secreted only by differentiated cells lead to the complete loss of tumor when you actually silence them? Okay, so the how do we answer that? This is secreted only by the bulk cells, which are not important for tumor initiation, right? So we use this particular paper where they showed, yes, of course, differentiated cells are not that important. However, in the context of tumor, they showed differentiated cells also play an important role in promoting the growth of the tumors which are initiated by stem cells. So this is an experiment they carried out. They actually injected stem cells alone, or stem cells and differentiated cells, and the differentiated cells alone, and stem cells and the fibroblast. So you can see when you inject only stem cells, they form a very small tumor. Okay, hardly you can see any bioluminescence. But they co-inject differentiated cells, you can see the same number of stem cells can form a bigger tumor. Okay, fibroblasts cannot do that help, and DGCs, differentiated cells, cannot form tumor by themselves. So basically, differentiated cells can actually help the stem cell initiated tumor to grow bigger. So the question we asked is, suppose we take DGCs in this model, silence them for FMOD expression, and then ask whether DGCs can actually cooperate, help the GSCs initiated tumor to grow bigger. So we did that. We used a murine model of DBT loop, and we can see that RNA level and protein level is more in DGCs compared to GSCs. And then we introduced this particular construct, which is an inducible shRNA construct under the control of doxycycline. And you can see here only in the DGCs, when you actually add doxycycline, RNA level comes down, protein level comes down. And then we took this GSCs, we are injecting GSCs, remember, where the FMOD expression is not there. Only when they become DGCs expression comes up, which can be silenced by this particular construct when you add doxycycline. So we injected GSCs, okay, and DGCs together. And then after 9 days, we added doxycycline to inhibit the expression of FMOD. These are the combination by which we carried out the experiments. You can see here GSC plus DGC containing non-targeting shRNA, GSC plus DGC containing FMOD targeting shRNA minus plus dox. So what is expected in this case, the non-targeting, whether you add doxycycline or not, there will be cooperation, tumor will be growing bigger. In the case of FMOD targeting, in the absence of doxycycline, there will be cooperation. In the presence of doxycycline, there will be cooperation initially before you add doxycycline. Afterwards, you should come down. Let's see whether it happened or not. You can see here GSC plus DGC with the NT, GSC with the DGC with the SHFMOD. And then initially, in the absence of doxycycline injection, all of them forming tumors, you can see the luminescence coming up in all the tumors. After 9 days, each of these two groups are divided into two groups further, and minus and plus dox. You can see on the left side, if you do bioluminescence, whether minus dox or plus dox, in the SHNT DGC co-injected cells, you can see the tumor continues to grow in both groups because you are not targeting any particular gene. And of course, the M-cherry comes up only when doxycycline added, because that is under the control of TRE. Whereas in this group where FMOD is silenced, when you actually inject doxycycline, you can see on the left side, in the opposite of doxycycline, tumor continues to grow. Whereas once you add doxycycline, FMOD gets silenced, the tumor growth decreases. Of course, these are controls. So essentially, to show in the quantitation model, if you see, DGCs are not forming any tumor, GSCs are forming a tumor, they're slowly showing the tumor at the end. This is the size they meet. But when you actually co-inject SHNT minus plus dox, the black and the purple, you can see that it takes off very nicely and forms a big tumor. SHF mod in the absence of doxycycline behave similar to these two groups, that is orange color, whereas the red color where after doxycycline addition, although there is initial cooperation and then it comes down similar to the DGCs. So meaning that FMOD silenced DGCs fail to support the tumors initiated by GSCs. So that means there should be some unknown function. Okay, FMOD secreted by DGCs are very important to support the tumor growth of the tumors initiated by GSCs. Of course, the function what we know so far is only it induces migration, doesn't affect the proliferation. There has to be some unknown functions, right? So based on our own studies as well as from the literature study, we somehow understood that it may have some angiogenesis inducing functions. We use this famous assay where you can promote the endothelial cells to form so-called angiogenic network. We essentially prove both FMOD derived from the conditioned media from the cells as well as a recombinant FMOD can actually induce the endothelial cells to form endothelial angiogenic network. So essentially we show that the FMOD can induce angiogenesis. Then we carried out a series of experiments and also a reverse phase protein array identified that there are two important proteins which are activated. HES1, which is a bona fide target of Notch signaling pathway, FAC, phosphorylated FAC, which is actually a downstream target of integrin signaling pathway, suggesting that Notch signaling as well as the integrin pathway are getting activated in endothelial cells when they are actually treated with the recombinant FMOD. To make the story very short, since I don't have a lot of time, so what we show in this set of experiments is secreted FMOD from DGCs bind to type 1 collagen to activate integrin, and then phosphofac and SRC, which activates the transcription factor KLF8, which in turn activates the JAG1, which is one of the ligands for Notch pathway, which activates the Notch pathway, leading to activation of HES1, which is an important target of Notch pathway, leading to HES has been shown to induce angiogenesis, leading to highly vascularized GBM. So essentially what we show here, alpha V beta 1, alpha 6 beta 1 integrin homodimers, heterodimers appear to be very important in this process, activating a FAC SRC pathway, leading to Notch HES1 activation. In that, JAG1 ligand appears to be important, and another linking molecule is a KLF transcription factor, leading to the activation of Notch signaling pathway. That's how it results in induction of angiogenesis. So why should you agree with me that this is really happening? So I will provide 5 evidences very quickly and then end my talk. Number 1. So if you take conditioned media from the stem cells as well as differentiated cells, both of them are able to induce the endothelial cells to form angiogenic networks. And that is a fact that the FMOD present in the DGCs is perhaps makes the conditioned media to induce angiogenic network formation. Is it really true? Yes, actually in this particular slide, if you see DGC conditioned media induced angiogenesis is actually compromised when you add anti-FMOD. Okay. And that can be rescued by addition of recombinant FMOD. So this is evidence 1 that FMOD actually can induce angiogenesis. Number 2, from a published paper where they showed there is a perivascular niche in the brain tumor stem cells. What they have done is in 4 different type of brain tumors, they actually stained for nestin plus cells. Nestin plus cells are actually the stem cells and nestin minus cells are the differentiated cells. And they tried to measure the distance of these 2 type of cells from the endothelial cells, which is stained by the MVD. Okay, this is actually one of the markers for the endothelial cells. And then they actually plotted percentage of tumor cells which are located as close as possible to the endothelial cells. You can see here, if you concentrate on the GBM, nestin plus cells are located very close to the endothelial cells. That makes sense. That means that the stem cells are having a very close association with the endothelial cells. However, the nestin minus cells, which are represented by DGCs, are also located very close, but lesser than the stem cells. Considering nestin minus cells, DGCs occupy bulk of the tumors, even though they are less in terms of proportion, because they occupy larger proportion of the tumor, DGCs are also, one can say, associated with the endothelial cells. Number 2. Number 3, is there any clinical relevance for our finding? You can look at in this data obtained from TCGA, FMOD, HES1, JAG1, all of them are upregulated in GBM. There is a positive correlation between FMOD and HES1, FMOD and JAG1. High levels of FMOD transcript predicts poor prognosis, and FMOD promoter hypomethylation predicts poor prognosis. Number 4, an experimental evidence from the lab, co-implantation experiment is not a good model because you are actually artificially injecting stem cells and the non-stem cells together from outside. That's not the way tumor actually evolve. Okay, you have a normal stem cell which forms a stem cell, cancer stem cell, and then it multiplies and also differentiates to form the bulk cells, and thereby the tumor is formed. Okay, so suppose during this formation of the non-stem cells or differentiated cells, if you silence FMOD using conditional silencing method, what would happen to the further growth of the tumor? That is a question we asked. We used 2 different murine glioma as well as 1 human glioma. And for this particular purpose, I'm going to show the data from only one animal model where AGR53 GSE, it's a murine glioma, where we used non-targeting as well as a FMOD targeting under doxycycline inducible construct for this particular experiment. AGR53 GSEs, again, transcript levels are higher in the DGC, protein level is higher in the DGC. Once you introduce this particular construct in the DGCs only when you actually add doxycycline, RNA comes down, protein comes down. So after verification, we carried out the experiment. The GSEs were injected, which are carrying this construct, and then after 10 days, the doxycycline was given, and multiple days the measurement was done for the M-cherry, which comes from this particular construct. So non-targeting as well as FMOD targeting. You can see early on, immediately after doxycycline injection, before silencing could occur, the tumor is there in both groups. Okay, FMOD targeting and NT targeting. Subsequent follow-up, we can see while the NT targeting, even though doxycycline is injected, the M-cherry continue to increase, the tumor growth increases because you are not targeting any inhibition in FMOD. Whereas on the right side, when FMOD is silenced, the tumor growth reduces and completely disappears. So you can show here in the form of quantitation, and also you can see a very big tumor on the left side, but not here, and also results in enhanced survival in the case where FMOD is silenced. And of course, FMOD levels are higher in the left side, but not in the right side. And FMOD secreted by de novo formed DGCs. When the tumor is being formed, when the stem cell differentiate to form the bulk cells, at that time, when you actually inhibit FMOD, the tumor stop growing subsequently. So that means FMOD secreted by de novo formed DGCs is needed for tumor growth. We wanted to check whether the angiogenesis is compromised because we showed in vitro that FMOD induces angiogenesis. That could be the reason why when FMOD is not there in the bulk cells, it results in the reduction in the tumor growth. If, of course, you can see the staining of EWF and CD31, there are 2 markers for endothelial cells. Both of them are highly present in the control condition, but not in the FMOD silence condition, which are shown in the form of a quantitative bar diagrams. You can see all the different markers shown in different kind of parameters. The angiogenesis is compromised in small tumors which are formed in FMOD silence conditions. And also another form of angiogenesis is vascular mimicry, where the tumor cells transdifferentiate to form endothelial cells. This kind of angiogenesis as well is actually compromised. You can see the GAP is actually a marker for the tumor cells. CD31 is the angiogenic marker, endothelial marker. There is a nice co-localization which is present in the control tumor, but not in the small tumor formed when FMOD is silenced. So essentially, both type of angiogenesis is inhibited in the absence of FMOD. So this is a model I want to end. Only 2 slides I have. Basically, in the tumor, you have a small proportion of GSEs, large proportion of DGCs. The FMOD is secreted only by DGCs, and we want to show that DGC secreted proteins like FMOD also play an important role in the growth of the tumor. We totally agree GSC alone can initiate a tumor. People have shown many times injection of DGCs by themselves do not form a tumor. Only GSCs form a tumor. However, in the context of the whole tumor, DGC secreted or expressed proteins like FMOD also play an important role in the growth of the tumor. So FMOD actually activates integrin dependent Notch signaling, and that results in formation of the highly vascularized GBM tumor. One additional last evidence I want to provide, if this model is true in the context of the endothelial cells, all these markers should co-localize. For example, FMOD and JAG1 and phosphofat and HES1 along with the endothelial marker like CD31. You can see here CD31 and FMOD, CD31 and phosphofat, CD31 and JAG1, CD31 and HES1. You can see a very good co-localization on the endothelial cells. So suggesting that indeed our model is actually working in vivo in the context of the tumor as well. And that indeed true, our model is actually happening in the context of the tumor. DG secretor protein like FMOD induces integrin dependent Notch pathway, leading to the formation of the highly vascularized GBM tumor. And finally, the take home message is the schematic which showed in the early on. Yes, of course, in the tumor, there are tumor cells, cancer stem cells, and differentiated bulk cells, in addition to the stromal cells. All of them are very important. While there is no question, cancer stem cell, most important thing, they are in the top of the hierarchy. People are trying to find all over the world to target cancer stem cells, but do not neglect bulk cells. Bulk cells also play an important role in the ultimate tumor growth, even though they cannot initiate a tumor by themselves, as in vitro animal models showed. However, during the tumor growth, the bulk cells also are very, very important in driving the growth of the tumors which are initiated by the cancer stem cells. So it is very important to get a durable response, to get a complete inhibition of the tumor growth. It is important to target not only cancer stem cell, bulk cells, as well as the stromal cells. So with that, I'll stop my talk. The students who did this work, Shiyoshi Sengupta is a graduate student who carried out the bulk of cells. Other student helped do this work, Arani and Prasasvi, and project assistant Mainak Mandal, and all my collaborators, particularly Philippe Marin from France, Cefifra, a project collaborator, and Professor Dino Friedman Morivinsky from Israel, who helped us to do animal model experiments and funding from various agencies. Thank you, and I will stop here. And I don't know, I'm okay with the time, or take more time.
Thank you, sir. Thank you very much. And then for your exciting and very reverberating talk. And I think many of the audience might have been benefited with this talk. And so, see, you have very nicely narrated and supporting with your own laboratory experiments that there is a trans differentiation can happen, and which will have a more impact on the tumor progression. So it was really a wonderful talk.
Thank you.
And so now the topic is open for the questions. If anybody has, please ask. There was one question from Kiran Kumar.
Yes, please.
Yeah, Kiran, please go ahead.
Yes, please. Kiran, please.
Kiran Kumar. Or Mahadev. Mahadev, if you are ready with your question, please go ahead.
Hello, sir. That was a nice talk.
Thank you.
Probably I must have missed it. Maybe you must have touched upon. Just wanted to know whether this FMOD directly has an impact on the endothelial cells and the vasculature formation, such as on matrix, etc.
We are not carried out such kind of assays. Basically, our assays are limited to having you have an endothelial cell. Different type of endothelial cells we have used. You have used HUX cells. We used lung-derived endothelial cells, lung tissue. We have used brain-derived endothelial cells, both human and murine origin. All type of as many as 4 different type of endothelial cells. The addition of FMOD can induce them to form angiogenic network. That appears to be the major methodology by which we demonstrate that in vitro FMOD can induce angiogenesis. In vivo, of course, we showed various data in the context of the tumor. FMOD appears to activate this pathway in the context of the endothelial cells in the tumor.
Thank you.
Thank you.
Kiran, you have a question?
I think that is wrong pressed. Can I have one question, sir?
Yeah,
Yes.
please.
Sir, looks like, is this hypoxia inducible?
The FMOD?
Yes, sir.
We have not tested that. We have not even thought about it. And of course, such kind of experiments can be done. We also work on hypoxia, but we have not looked at in that direction.
And a small basic question. Sometimes I get confused.
Yes, please.
The difference between cancer stem cell versus metastasizing cells, like transdifferentiation cells.
Yes.
So how we can distinguish a little?
Okay, I will tell you whatever I know. Of course, the metastasis is something which does not happen for the brain tumor cells. The primary tumor of the brains are the glioma, where they do not metastasize out of the brain. Okay, unlike other cancers like breast cancer, that is something. However, the glioma tumors are highly notorious. They can move from one part of the brain to another part of the brain. Okay, within the brain. Okay, which can be considered some kind of equivalence of metastasis. Okay, but in the context of the brain, where breast tumor, I have listened to many people, including Bob Weinberg and their postdoc, Professor Sendurai Mani, and many people. The EMT, the process of EMT is connected to creating the metastatic cell. However, the phenotype, the epigenetic changes which are occurring when the cell undergoes EMT is largely similar to the stem cells. Okay, so cells which undergo EMT, once they become EMT, the changes occurring in the gene expression largely similar to what is happening in the stem cells. So in principle, they are not able to differentiate between the stem cells and EMT undergone cells. Okay, so it's very difficult to answer whether stem cells are the one which undergoes metastasis or the ones which undergone EMT, because they look similar to each other. There is now new concept. There is something called in between, MET and EMT, and in between there is some kind of a compromised cells, both type of behavior. They appear to be the most going towards the metastasis. It's not very clear, but definitely stem cells play a major role in the process of metastasis or invasion otherwise.
Thank you, sir.
I have a question, sir.
Yes, Brahmananda.
Sir,
Yes.
this FMOD is what is the normal function of FMOD in the normal cells? And that is my first question.
Yes.
And the second question is that is this FMOD is very specific to the brain tumors, and its role in the other tumors has been elucidated.
So FMOD actually, there is a class of this proline-rich protein, like extracellular proteoglycan. There are a series of proteins are there, bunch of proteins are there in the group, and most of them are upregulated in many cancers, including FMOD. Okay, so it is present in the extracellular space. It is secreted protein, and from our studies and few other studies, people show that it induces a migration and invasion in vitro assays. Okay, it does not seem to affect the cell proliferation. If you overexpress or silence this protein, cell proliferation is not affected. Okay. However, the interesting part is when you silence, it affects the tumor growth. That is what actually resulted in our study, that induces angiogenesis in addition to migration inducing function. Thereby, we found an important role for bulk cells in the tumor growth.
So is it published, sir? This work is published?
We are trying to publish it. Yeah.
Okay.
We are just,
so there's one more question. I think someone a present diagnostics is the present diagnosed system able to distinguishable currently all the time, or there is a possible to mislead the test results? It's a general question. That's what they ask.
I didn't understand the question. What was that?
So the present diagnosis system is able to distinguish correctly all the time, or there is a possible to mislead the test results.
So you mean for example, in the context of glioma, you have different grades, grade 2, grade 3, grade 4. The current methods available, is it very accurate to identify which grade of glioma it is? What is that the question they are asking?
It's something
I'm Dr.
Yes.
Nagaman, Institute of Science Department.
Yes, madam. Yes.
Sir, my as a continuation of Bindu Madhav sir question, I just asked this thing, because the variation of these cells, this cancer cells and normal cells, is it possible distinguishable? How good the current technology of medical science is helping to distinguish kind of thing?
I guess the question you are asking, madam, is I talked about stem cells and the non-cancer stem cells. How these cells are identified? Of course, in vitro in the lab, when you get a freshly resected tumor, you can make single cell suspension and divide them based on membrane markers by flow sorting. Okay. However, in the clinical settings, you actually get a tumor and then you make sections in the pathology department. How do you identify stem cells versus non-stem cells? Okay. So one best way, of course, everybody does this. You can use immunohistochemical approach to identify percentage positive T cells, which are specifically expressed markers, which are expressed only in the stem cells versus non-stem cells. Thereby, one can actually identify proportion of stem cells and non-stem cells. And perhaps one can also use that to using techniques like laser capture dissections like that to separate out the stem cells and non-cancer stem cells. Is that answered your question?
Yes, sir. Thank you. Thank you very much, sir.
Thank you.
Your talk was very nice.
Thank you very much.
Any more questions? It looks like there are no more questions, sir.
Okay.
Once again, thank you so much for for joining with us. I think and a big round of applause to you. I think
Thank you.
unlike-
Thank you.
Thank you.
Thank
so
you.
much.
I hope you all enjoyed and
Yes, sir.
it
Yes,
made
we
some
truly
sense.
enjoyed. Yeah,
Bye
we
bye.
truly-
Bye bye. Thank
Bye.
you. Yes.
Thank you, sir. And we have now the next speaker, well-known clinician in and around Hyderabad. Many of his students are in several hospitals serving patient care. He is none other than Dr. Raghunath Rao, D. Raghunath Rao. Sir is a clinician, as I said, MBBS, MD, and DM. And to go by his long list of accomplishments, so sir is currently at chief medical oncologist at KIMS Icon Hospital. So this is the Tata Memorial Hospital, Mumbai, funded by Department of Atomic Energy, has given him a charge to establish same hospital in Visakhapatnam. Sir is one who is established there and then acting as a chief medical oncologist there. And to read about some of his accomplishments. So he's an elected fellow of American College of Physicians for the year 2020 and Royal College of Physicians of London 2019. And he is the elected fellow of Andhra Pradesh Academy of Sciences. Presently, he is the president for that academy as well. And sir also been awarded with the Dr. B.C. Roy National Award for Excellence in Promotion of Oncology by Medical Council of India, and a national coordinator for training government doctors in cancer screening and prevention. And he is one of the collaborators for the Breast International Group, Usmania University, Central University, NIN, CCMB, etc. And established first population-based cancer registry for Hyderabad, and established first hospital-based cancer registry in Andhra Pradesh, and first hematopoietic stem cell transplantation unit in NIMS, Hyderabad. I think sir is there in everywhere, the first. And started at Twin Cities Cancer Club in 2004, and first ACRP certified clinical research physician in India, and started a course, DM, medical oncology course in NIMS back in 2005, and also founded the Medical Oncologist Forum for the state in same year 2005, and started a childhood cancer study group in 2007, established a subsidized cancer pharmacy at NIMS. That's where the pure patients will definitely benefit, way back in 1992 itself, and established a clinical research facility in NIMS, where we are also collaborating now. We are enjoying those fruits. And he is a pioneer in phase 1 clinical trials in cancer drugs, and he's been part of over 80 clinical trials since in 1993. And first medical oncology department and comprehensive cancer care center for the state of Andhra Pradesh at NIMS. So sir is is actively involved in establishing the center at Vizag. So he's the one who started the oncology department in NIMS in 1989 itself. So, and not only this, he is active in research, published as a co-author for more than 280 publications, and then part of several research grants. And we are very glad to have you in this talk, sir. And thank you very much for taking your precious time and then sparing with us, and then also educating some of the clinical aspects and your experiences for the audience. So now I would invite you.
Thank you very much for the invitation, sir. Thank you for the very generous introduction. My talk today is essentially on a clinical aspects. As I said, it is completely something which you have developed, say, in a university, and then it comes to the bedside of the patient. So I shall take you through the advances in the systemic therapy of cancer. You heard the Vice Chancellor and Professor Somsundaram elaborate on what is the basis, what is the Darwinian theory as applied to evolution of cancer, what is the survival advantage of a cancer cell, and how does it exploit. Professor Somasundaram has told you very clearly that it is not just the cancer cell, but the bystander cells or the milieu or what we call the area in which the cancer is growing. So the soil is equally important rather than the seed. So let us have a look at all these aspects as they translate into clinical medicine. So this is in a short the summary of the epochal turning points in modern oncology. We knew for a long time that many great surgeons and physicians have treated cancer all the way to the late 19th century using surgical techniques. In fact, the so-called first principle of Halstedian.
Sir, you have to share the slides if you are presenting.
I'm sharing the slides, sir.
We haven't got yet.
It's actually shared.
No, press first press the button, share button on the window.
I am doing that only. I'm sharing my entire screen. Okay. Share your contents of your screen. Now I'm not getting the share button. Is there any other way of doing that? I can see. I'm asking it to share, but it is not.
Sir, which email ID that you are using it? Use the same email ID, otherwise it won't accept it.
I'm only on one email ID, sir.
So usually that is what happens. I think one of the
I logged in with the same email ID.
And and then admin has admitted through the same email ID or some different email ID.
Same email ID, sir.
Sir is activated.
Maybe you log out once again and then log in, sir. That would solve you.
Okay, let me log off once again.
Because sometimes PPTs are open before you join, so they may not accept it.
Okay, let me see if I need to log in again. Okay.
And also close your PPTs also.
You want me to close it once?
Yeah, close the PPTs because you open the PPTs once after joining.
Okay, sir. So let me go back to the email. I should first log off, you said. I'll try to log off. I will leave and then log in, sir.
Yes, sir. Yeah.
I have joined back, sir.
Yes, sir. Now try again now.
Okay, I will try again.
Kiran, you want to say something?
No, that's
It's not allowing me to share my entire screen.
I think Bindu, probably you can take the sir. Please you share the slides to Bindu.
I don't have that. Sir to sir. Okay.
Sir, is that the upper arrow icon is not activated?
It is activated, sir, but it says.
Just click that, sir. Click that.
1 second. I'm asking it
Up
to.
arrow icon.
I'm asking it to share a tab at least. Let's see, sir, whether it will share a
Tab
tab.
and then down button you have to say yes once it pops up, once it is enabled, the button.
Yeah, I will try that again. I said a tab. It is not showing my screen. It is not showing my screen. But
Or select entire window itself, sir.
I'll
And
select
click on
it.
the window again, sir.
Yeah, a window. Okay, I will select now. Okay, sir, I got it.
Yeah.
Okay.
Now
I got it. Thank you very much. It was not willing to allow me to go through it.
Yeah. Okay.
So I'm so sorry for that glitch.
That's fine.
Okay, sir.
Now we can see, sir.
Yeah.
These are the turning points in modern oncology. We first had surgery alone as the treatment of choice. From there, we went to radiation therapy. And sometime during the mid 1900s, that is in the mid 20th century, chemotherapy started playing a role as systemic therapy for cancer. As you know, surgery and radiation therapy are essentially parts of treatment for localized disease. They are not effective for systemic treatment of a cancer. So there are two theories in cancer. One, that even when the tumor is very small, that the tumor cells leave the parent organ or the place from where the cancer has arisen, and they disseminate far and wide, waiting for a chance to grow. So the milieu is pretty important. So they may show up several years later or several decades later. So the concept that a total cure is possible only through systemic treatment took root in that the mid 20th century. Soon after that, we had some insights into the molecular basis of cancer. Till then, we were dependent on clinical, epidemiological, as well as pathological features of a cancer. We just looked at a tissue section or a cytology smear and said this is cancer. So we understood what's the basis for cancer, what happens within the cancer cell, a little bit, if not everything. And then we started targeting treatments. And then we went on to develop several other treatments down the line. So if I concentrate only on chemotherapy as the systemic treatment for cancer, then we started with animal models in early 20th century, that is 1900s, with arsenicals. Then we developed, the most important part was the experimental setup where we had transplantable tumors. So you transplanted tumors into immunodeficient mice and then used systemic treatments on the mice to know which of the treatments are likely to be effective. The first treatment which was found to be effective was in early 1935, soon after the First World War and before the Second World War. We used nitrogen mustard as a gas in the treatment. We discovered that in the autopsies of those who died from inhalation of nitrogen mustard, that the entire lymphoreticular system was depleted in the body. So it was used successfully as a treatment for a lymphoma. Well, we soon realized that it is a short-lived response, but indeed it is a response. And then we developed several cancer chemotherapy national service centers around the world where you used several different anti-metabolites like antifolates, thiopurines, methotrexate as treatments. And the first tumor to have been cured completely happens to be choriocarcinoma, which was a gestational trophoblastic tumor. In fact, the man who discovered this in the National Cancer Institute in the United States was asked by his superiors to desist from saying that he can cure cancer. And when he persisted in publication of this article, he was asked to leave the National Cancer Institute in North America. So with that, we come to the first publication of choriocarcinoma as a concept for cure, and which was embraced by several others as a part. Now, the governments now pitched in, and then we could start using collaborative groups. That means the same type of treatment at different centers, the same protocol was started in different centers. We learned that we can cure acute lymphoblastic leukemia and Hodgkin's lymphoma. And then, as we said, we went on to cure the fourth cancer, that was testicular cancer. The concept of an adjuvant treatment, that is, you remove the bulk of the tumor through surgery, and then for whatever is not visible to any laboratory test or to the naked eye, you use adjuvant treatment to root. Then the National Cancer Institute started investing in molecular biology as a tool to see what happens to the cancer cell within the cell and as it interacts with other cells. Soon, we started seeing a decline in mortality for cancer in the late 20th century, and then we developed in vitro systems to test many a drug or in silico experimentation. So once the genome was sequenced and then the molecular profile of various cancers vis-a-vis the normal cell was made available, the rapidity with which the drug discovery started was become explosive. So you have an explosion of many different kinds of drugs targeting anything other than chemotherapy. Now, why does chemotherapy fail? Part of this was explained by Dr. Somasundaram and our Vice Chancellor. You have a tumor, you consider that the tumor to be a uniform group of cancer cells. No, they are not. There are already different types of cells, A and B, and you try to target the largest population of these cells or the what we call the druggable component of this tumor. And if you are successful, your target works and majority of the cells die, then there may be no residual disease. But more often than not, two things happen in the tumor, rightly explained by my predecessors. One, it is pretty heterogeneous. Not all the cancer cells are sensitive to the same drug or groups of drugs. Two, the treatment you have given actually induces a resistance in the tumor, so it acquires. So it is treatment-driven resistance which actually leads to a recrudescence of the disease. Now, it is very improper to think that the disease which has occurred, although morphologically looks like an adenocarcinoma or squamous carcinoma, is the same disease as the original tumor. No. Molecular profile wise, typically, it may have, as you rightly know, acquired several other very useful mutations, which was very clearly explained by the Darwinian hypothesis. And then you get a completely different molecular makeup of this tumor. So you once again treat the tumor with either conventional chemotherapy or targeted therapy. If you are lucky, you will end up with no residual disease. If you are not so lucky, you will have a recrudescence of the disease. You try again and again and again. And finally, the patient succumbs to progressive disease. So this is typically what we find in a cancer. We now come to the era of the NIBs and the MAPs. The NIBs are those which target simply those which are called small molecules, chemically synthesized in the laboratory, which are highly effective in preventing a tyrosine kinase activity. So that means downstream signaling pathways can be stopped. On the cell surface, you find many a receptor or a ligand to a receptor against which you can develop a monoclonal antibody. For example, here I have listed bevacizumab, aflibercept, cetuximab, panitumumab, trastuzumab, ramucirumab. The ones on the right, that is cetuximab, panitumumab, all of them work on the same receptor superfamily, that is the epidermal growth factor receptor superfamily. As opposed to that, bevacizumab works on something which Professor Somasundaram has alluded to. It works on the milieu. You know that cancer cells are very adept at inducing neovascularization. new blood formation. In fact, cancer cells themselves can line up as tubes and then start getting feeded. They need it for two reasons. One, to get nourishment. Two, to start migrating. That means they use the same pathway as the tubes to migrate out of the primary tumor. So you can have a monoclonal antibody either against the growth factor or against the growth factor receptor, or when you come down to the receptor's activity. So you have receptor tyrosine kinase is activated, and then downstream signaling pathway starts. So you can block any of the downstream signaling pathways using a variety of tyrosine kinase inhibitors, which we call NIPs. So NAPs or NIP. So the others are MAPs. These are called NIP. So crizotinib, ceritinib, alectinib, imatinib, all of them are drugs which act at downstream signaling pathway, switching off the pathway, and then restoring some semblance of normalcy in the cell. So molecular targeted therapy has come a long way into almost perfection, to the extent that now, at least in certain diseases like chronic myeloid leukemia with BCR-ABL oncogene alteration or a fusion oncogene being formed, you can now think of stopping treatment for patients who can no longer have a detectable disease. Well, the term detectable disease is a euphemism. So the detection is, of course, as sensitive as your assay system, nothing more than that. So whether it's a PET scan or a nested PCR, if you cannot detect a tumor, your assumption is that there are no detectable cells. But that may not be the truth. As rightly pointed out time and again, that you can have a very quiescent stem cell which cannot be picked up by any test whatsoever at the moment. So it's important to understand that we may actually have drugs which are targeting cell proliferation and survival either within the extracellular matrix or through the intracellular area or cytosolic area in the form of several monoclonal antibodies or tyrosine kinase inhibitors. It is also important to understand that the milieu or the blood vessels themselves can be targeted both outside the cell membrane and within the cell membrane. For example, sorafenib, which is a multi-kinase targeting agent, can actually target many tyrosine kinases at the same time, one of which is, of course, the VEGFR1 and VEGFR2 downstream signaling pathways. So we have seen that the first era of molecularly targeted therapies had NIPs and MAPs as the drugs of choice. Now we have something called antibody drug conjugates. So you remember that we used in the past several chemotherapy drugs which were toxic to the cells. The chemotherapy drug, of course, is not very selective. It will divide any of the rapidly dividing cells, like those which form your skin, the lining of the mouth, all the way to the through the gut to the anus, the hematopoietic stem cells and the lymphopoietic stem cells. So that's the reason you see neutropenias, anemia, thrombocytopenia, hair fall, mucositis as a side effect of chemotherapy. But instead of that indiscriminate action of chemotherapy, if you can link the chemotherapy drug to a specific target, like using a monoclonal antibody, which we have studied in the past, which are extraordinarily highly effective, say in breast cancer or in lymphoma, rituximab and trastuzumab or bevacizumab, then possibly you can avoid the so-called innocent bystander effect of the drug. So that is where antibody drug conjugates come in. Whereas the antibody uses the natural killer system to produce antibody dependent cell mediated using the lysosomal system, and then can either stop the signal transduction or can arrest the cell in mitotic phase through a microtubulin polymerization. For example, in TDM1, which is the drug, which is chemotherapy drug, which is used is M-tansin. M-tansin by itself, if you give to human beings, is phenomenally toxic. So the amount of drug which is used for conjugation is so low that it's about 1/200th of the standard dose, but still it is extraordinarily effective in those tumors which are HER2/neu positive in breast cancers. So you have a dual activity, and at the same time you avoid toxicity to the normal cells. So here we are trying to refine the so-called precision medicine using two drugs which are independently active but can be toxic, but have a synergistic action to kill a cancer cell by two different means. Next we come to, you heard this checkpoint, but the checkpoint which our Vice Chancellor had mentioned is about the mitotic checkpoint. The checkpoint we are interested is in the energy induced by the tumor cell against the recognition by the antigen presenting cell and in turn by the T cell, which is exploited by the tumor cell. So we know that there is an immunopoiesis in cancer, that the immunocompetent cells, that is the antigen presenting cells and the T cells in the body, can actually do not recognize the existence of the tumor cell because of the blockade induced in the T cell after the locking of the major histocompatibility complex with the T cell receptor through the PD-L1 and antibody and the PD-1 antagonism. Similarly, the T cell CTLA-4 antibody, the receptor actually competes with the B7 of the antigen presenting cell. So both of them actually block the T cell from recognizing the tumor cell. So in order to overcome this block, you can either block the CTLA-4 using ipilimumab or tremelimumab, so that the antigen presenting cell now processes the tumor antigens and presents them to T cell to induce an endogenous response or to induce an endogenous cell kill. On the other hand, you can use monoclonal antibodies like pembrolizumab, nivolumab, or atezolizumab, durvalumab, and avelumab to overcome the resistance or overcome the interlocking of PD-L1 and PD-1. So the ligand with the receptor blocking can now be overcome, and then the T cell will interact with the tumor cell and induce a natural killer cell activity. So far we have seen many different varieties of immunotherapy in cancer, but we need to know that there are many multiple regulatory pathways for the tumor to evade the immune system, and these pathways appear to be non-redundant and hence you have a possibility of enhancing endogenous immune response by therapeutic blockade of these pathways. Unfortunately, majority of the solid tumors do not mount an adequate endogenous immune response, and hence response from immune checkpoint inhibitors, if at all, is very modest. In some tumors, it's very dramatic. For example, if you use in melanoma, ipilimumab combinations or nivolumab combinations or a combination of both, that is the blockade with the antigen presenting cell as well as the tumor cell, combinations can produce a dramatic response. So we now need novel anti-cancer immunotherapies which can work across several tumor types. So irrespective of the tumor, it can work. For example, nivolumab can work not only in Hodgkin's lymphoma, it can work in, say, a gastric carcinoma, it can work in a melanoma and several other tumor types as well. So Unfortunately, there is, with the continued use of the drug, a mechanism by which the epitope is lost or suppressed, and then the immune response can be completely lost. So that means you need to understand that even now with immunotherapy, if you use it later and later in the course of the treatment of a tumor rather than earlier, you will start losing an unsustainable response. Now we come to the CAR T cells. CAR T cells are T cells in which a genetically modified or a gene is introduced, which now can actually recognize through its ectocellular domain the cancer cell. So I told you that there is an energy which is induced, or an immunoperesis which is induced by the cancer cell. So the T cell fails to recognize that there is a cancer cell wherever it is. In fact, tumors are very rich in lymphocytes, but yet they are not attacking the tumor cell. So you need to induce a genetic change in the T cell to start recognizing a bit of the tumor or an antigen on the tumor, an epitope on the tumor, a specific tumor, and then synthesize them in vitro. So this is pure genetic engineering, where you remove the cells of the patient and then transfer them to a petri dish, and then to use a genetically engineering technique. I'm not going into the details now, and then introduce a gene which is expressed on T cells, expand the T cell clone, make them into a sizable amount of number of T cells, say in a millions, and then transfuse them back into the tumor, into the patient with the tumor, and suddenly the tumor can overnight disappear from the patient. It is actually very dramatic to see this response. It is so very heartening to see this response, whom you have given up as no treatment available in future. So we now have engineered CAR T cells, the fourth generation of CAR T cells. It only depends upon the number of downstream pathways it is going to stimulate, or the number of pathways it is going to recruit in the immune process, immunologically driven tissue destruction. So you select the patient cells through leukapheresis and then elute the cells either in a positive or a negative manner, expand the cells, use a transduction, whatever is the method you want to use for transduction of the new gene you want to introduce into the T cell, use that, expand it, then remove the cells from the, harvest them back from the system or the petri dish or the flask. Here it's a flask. Wash the T cells, formulate them, and then put them back into the human being. So this system of treatment has worked dramatically well for several types of hemopoietic cancers, like childhood leukemias, adult leukemias, multiple myeloma, you name it. But remember, each one of this is patient specific. Now technologies are now available, being made available. Soon, by the end of another couple of years, we will be able to have something which can be infused into any given patient. So you will have the major histocompatibility loci which are masked from any given patient, but only the epitope is expressed, and you have T cells which can work across different human beings. So this is the level to which you can use genetic engineering to expand T cells in vitro and then transfuse them in vivo. So the day is not far off when you can actually buy it off the shelf. Right now, it takes you about 6 weeks and $1.5 million to prepare a CAR T cell preparation for an individual patient. So this is an individually driven sort of treatment. As opposed to that, I told you that you can use a payload for a given monoclonal antibody. The payload I had mentioned was a drug, a chemotherapy agent. Instead, you can use a radioisotope also. So you tag a radioisotope to that drug, and the radioisotope can also be taken up by the cell. Here is an example of a clinical patient of a terminally ill Hodgkin's lymphoma patient who, within just 5 weeks of treatment, has no evidence of the Hodgkin's lymphoma at all. So that means you have a monoclonal antibody to which you tag a radio tracer that can be very highly effective in treatment. You can use any sort of radioisotope. You can use a beta particle or electron. You can use an alpha emitter, or you can use an Auger electron like iodine with different half-lives. The half-lives can be as short as 46 minutes to as long as 60 days for radioiodine. So you can use many different varieties of radioisotopes for tagging along to a monoclonal antibody and injecting them. The beauty of this treatment is you can take an exposure at say every day or every week or every month, because the tracer is still concentrated in the tumor, and you get something like an auto radiograph of this patient responding to the tumor. Along with the tracer, the tumor will also disappear. So I have told you, I have listed here some of the monoclonal antibodies which are now in clinical use, to which a tagging has been done. The radioisotopes used have been mentioned here. So you can use it in non-Hodgkin's lymphoma, acute myeloid leukemia, colorectal cancer, GI cancer, breast cancer, ovarian cancer, prostate cancer, myeloma, etc. So many different varieties. Another form of systemic treatment which we can exploit is what is called synthetic lethality. For example, in panel A, I have shown you a normal cell to which you give a PARP inhibitor. There is absolutely no effect. So a normal cell which doesn't carry a BRCA mutated gene has no effect whatsoever with this drug. But if the cell has a BRCA mutation and is unable to do a DNA, undergo a repair done to its DNA, either because of a chemotherapy agent, or because of radiation, or because of a natural process of tumorigenesis, suddenly becomes now highly susceptible to a second agent which interferes with the same repair mechanism. So downstream repair mechanism. So imagine that you have a drug which is used with a BRCA mutant cancer cell, and now you can exploit the inherent nature of that cancer cell's BRCA mutation to see that you produce an additional synthetic lethality from the bench to bedside. So this is the way you have developed olaparib, rucaparib, and niraparib in the clinic, and it is highly effective because we now know that you do not need to have an inherited BRCA mutation for this treatment to be effective. BRCA mutations indeed are as common as from 15 to 25% in as varied tumors as prostate cancer to GI tumors to lymphomas to hematopoietic cells. It need not be restricted to a breast cancer at all. So it looks like sporadic BRCA mutations are much more common than what we actually ascribe them to. So it is important to test the tumor for the BRCA mutation as opposed to looking for a germline mutation in a hereditary breast ovarian cancer syndrome. There are other ways of exploiting this lethality, for example, using PRMT5 inhibitors, which can cooperate with the MTA and induce a cell death. So many ways of using a the same enzyme systems which repair, use the same substrate, and then you can use a DNA repair enzyme system deficiency to your advantage in the tumor. So we now have a hugely expanded armamentarium for systemic treatment of cancers. I have not touched the cancer vaccines in this. That is a completely different way of doing with it. We used cytokines. In the past, for example, we used interferons and interleukins. We are still using interferons and interleukins in a different fashion. I told you about adaptive cell transfer using CAR T cell therapy or dendritic cell therapy. I told you about immune checkpoint inhibitors and, of course, some form of synthetic lethality for the cancer. In the center is what is depicted through the idea which Professor Somasundaram has told you, that it is important not just to concentrate on the cancer cell or the cancer stem cell, but the milieu in which it is growing. So that is equally important as well. So everything needs to be looked at in total. So this is the third edition of the Hallmarks of Cancer. The first edition was formed in the early 1990s. The second edition came in the year 2011, and this is the edition of 2022 with ever expanding idea about the molecular basis of cancers. We now know, apart from what has been given here, that is evading growth suppression, avoiding immune destruction, enabling replicative immortality or tumor promotion in inflammation, etc., resisting cell death or apoptosis, sustaining proliferative signals, we have added 4 more. One is unblocking the phenotypic plasticity. Some part of it was touched in the glioblastoma lecture. Okay, these are called emerging hallmarks and which need to be exploited in the laboratory and brought to the clinic. Non-mutational epigenetic reprogramming. So we have exploited this to a certain extent using acetylators and methylators. So you know that a gene can be silenced by adding methyl groups to it, or a gene can be activated by adding an acetyl group to it. Both forms of treatment are now available in the clinic. For example, we use decitabine in myelodysplastic syndromes to treat patients with elderly patients with acute myeloid leukemia as well. Then we also have some idea of senescent cells and what they do, and although not there fully yet, and we are just about to take notice of the larger picture away from the tumor, that is the polymorphic microbiomes which live on every surface of the human body and within the gut as well. So this is what I said about unblocking the phenotypic plasticity. We know that there is a plasticity with which the cell can dedifferentiate or can transdifferentiate. It is what we call the epithelial to mesenchymal transition. So you can block the differentiation, or you can restore the normal differentiation back to the cell. So this is what is we call unlocking the phenotypic plasticity. Many non-mutational epigenetic reprogramming pathways are now available. We are exploiting them in the clinic already, but we need to use them to a greater degree so that without the altering the genome of the cell, you can actually use the epigenetic reprogramming of the cell. I come back to one of the most important topics now, hot topic is polymorphic microbiomes in the cell. At least in the gut we know, and far away from the gut we know, it is not just cancer. They seem to be involved in many other chronic disorders, including diabetes, hypertension, heart disease, Alzheimer's, and your just aging phenomenon, or things like psoriasis and lichen planus. So they modulate growth, inflammation, immune evasion, genomic instability, and treatment resistance. So the gut, the lung, the skin, the vaginal mucous membrane, the cervical epithelium, in conjunction with the tumor, seem to matter quite a lot. And for the senescent cell as well, the senescent cells can be of different origins. They seem to quietly stay there, and when the time comes for them, they seem to be able to promote cell growth. At least we know that the milieu is very important in the metastatic bone disease. For example, we now use a rank ligand antibody or rank ligand anti-rank ligand drugs to block the metastatic disease from eating away at the bone. It is actually the normal cell which is providing the growth factor and the pathway for the lysis of the bone to form a lytic tumor, so that there is a niche created for the tumor to grow. So it is creating a space. It is created by the normal cell itself. And the normal cell actually contributes, the osteoblast actually contributes to the tumor growth. So it is important to understand that the soil is as important as the tumor. So that leads us to the new additions which I have mentioned. There may be some more which will soon start getting into these new additions. And for us, the future is to move from trying to find a cure to preventing cancer. And I'm sure the gut microbiome will be one of the pathways through which we will be able to prevent not just cancer, but many other diseases as well. So instead of imagining a tumor or metastasis as distinct from the individual, you need to understand that everything in the body, every system in the body, which you target, which you don't target, actually interacts with the tumor and either inhibits the tumor or promotes the tumor. So it is not independent of the organism as we are led to imagine. So there is an interplay between inflammation, primary tumor and the metastasis, between thrombosis and the neuroendocrine changes, between cachexia, metabolic changes and the immune inhibition. This is very well documented now. I know Professor Redana is on this conference, who has worked with global inflammation and carcinogenesis. Yes, that is the truth. So you can actually view it from that angle, and we can make a guess that yes, COX inhibitors are extraordinarily good anti-cancer agents, at least in the preventive phase. We know the famous aspirin trial, which prevents colorectal cancer. Now the evidence is actually gathering that the vegetarian diet is phenomenally rich in acetyl salicylic acid, and that's the reason why the vegetarians are less prone to develop colorectal cancers. As they migrate from India to say Britain or Canada, and the next generation stops eating the Indian diet and then goes on to develop a Western diet, we or eats a burger and a pizza, we notice, or a hot dog, we notice that this vegetarian component of the diet goes down and the meat component actually increases. And we know now that has been proved by migrant population studies. We did a study with Oxford, which is the largest study in the world, which shows that migrating populations, these generations, which change over from their diet, which is largely plant-based to meat-based, actually incur the new set of tumors which are common to that native population of the country which they adopted. So, ladies and gentlemen, it's important to understand that the cancer cell is no longer in isolation. It is part of your system. It is exploiting the system. It is exploiting every organ system in the body, and the organ systems are either mute spectators or they are actively participating in the promotion of the cancer. So the field is out there, ripe out there for each one of you to exploit and find out how each one contributes and how we can exploit, either by turning off or putting them into overdrive. So it's important that You need to join hands to close this care gap. If we were to get all these into the Indian population, it is the universities which need to actually ramp up their innovative minds. That means the scientist now must become the innovator, and the innovator must now translate it into a product which is now medicinally active. This is the gist of my talk of systemic advances in systemic therapy of cancer. So today, all of you can take a pledge to move our science from the lab to the clinic. Thank you very much for your patient hearing.
Thank you very much, sir. And sorry, I think we strained your throat. So, nice talk. Shared a lot of clinical aspects, and that is interesting that migratory population who changing from vegetarian to non-vegetarian correlates with more risk of cancer. It's a nice, sir. So I'll take any questions from the audience if there are any.
I have a question related. Hello, sir.
Yeah, please, I can hear you.
Wonderful talk, sir. Like, a lot of, clinical aspect that we realize, although, we are basic scientists, many things we know about the basics, but when it will try to translate, many times that we do not understand those concepts properly. And so see the new technology of this immunotherapy, CAR T cells. So how effective is that? Is it applicable to all the cancers, particularly the breast cancer? Because I work on that, so I would like to know if the CAR T cells can be applied to the breast cancer.
Sir, no cancer is so difficult that it cannot be exploited through CAR T cell therapy, sir.
Okay.
So to be very frank, yesterday's articles actually all over reported that the longest survivor of a leukemia who relapsed, 2 patients were studied. After transplantation for a leukemia, 11 years is a patient's 2 patients of CAR T cell therapy. Lymphatic leukemia, the other is an acute myeloid leukemia child. This is 11 years from their treatment. So people actually studied to see what is it that makes their tumors completely disappear. So the problem appears to be that the T cells which you have put into the system start losing their effectiveness. So you need to allow them to integrate very comfortably into the host immune system and continue to express the activity within the system. So down the line, 1 or 2 years down the line, if they stop expressing and there are still some stem cells left behind, the cancer will come back. So technically, it is feasible to raise a CAR T cell against any cancer. The imagination is what is the epitope you want to raise it against, and will the cancer cell continue to express this epitope with great fidelity down the line? If it shuts off the epitope, the CAR T cell you have prepared is of no use to the body. That is the problem. So some amount of fidelity is required, sir.
Thank you, sir. Thank you very much. Yeah.
Bindu, can I ask one question?
Yeah, Dr. Mahadev.
Hello, sir. That was a nice talk. In fact, along the same line, what Dr. Brahmanandam asked regarding the CAR T therapy. I know it is very promising, and in fact, as you said, it costs millions of dollars or something like that. So the country like ours, like where the population is high and the poor people, can we really, how robust is going to be? And can we see a light? How soon it will be? You think?
Our country is poor in financial resources. It is not poor in intellectual capabilities. This
no, it's not about the intellectual capabilities.
Tell everyone. Any one of the CAR T cell labs you go anywhere in the world, there is an Indian brain, possibly somebody from your own city who is working in that CAR T cell lab. This is the absolute truth. The same thing happened with chemotherapy drugs. Every single one of the giants, for example, in Basel, the 5 giants were full of pharmaceutical chemists and scientists from India. We supplied them to the rest of the world. Likewise, every single monoclonal antibody, NIP and MAP, has some Indian behind it in the manufacturing process. So, what is the problem with India is that the intellectual wealth we are not allowing it to innovate and invest within India. If we can turn, for example, rituximab, when it was manufactured and supplied in India, the original cost of the drug was roughly 1,85,000. When it was manufactured in India and which is supplied in India for the first time by Dr. Reddy's, the cost dropped down to 25,000 rupees. Now rituximab is available in India for 8,000 rupees. So we can actually compete with the rest of the world with our technology, with our manufacturing capability, and our innovative approach in pharmaceutical sciences. The idea is to bring down the cost through synergism. How many universities have invested in CAR T cell? Only university which has invested in a synergy is IIT Bombay with Actrec of Tata Memorial Hospital. Others have not invested. Why not? Why can't they? Every single one of these universities invest in innovation which translate into clinically meaningful drugs? Yes, it is possible. Because I'm very sorry to say that the intellectual property all over the world, at least in pharmaceutical sciences and medicine, belongs to Indians. It doesn't belong to, at least in the manufacturing process, it doesn't belong to any Westerner. The concept may have been theirs, but the way we bring down the cost is unbelievable. So, if you start manufacturing CAR T cell therapies in India, I am sure instead of 1.5 million, it may be something like 1.5 lakh rupees. So, from 15 lakhs, you may get it to 1.5 lakhs. Now, the Andhra Pradesh government and Telangana government actually support the transplantation. You can do an auto transplant for 8 lakh rupees and you can do an allo transplant for 12 lakh rupees. So if you open up this, you can actually see the potential for doing. In Hyderabad, there are 28 centers which can do a transplant now. Okay, so it is actually a way of lowering the cost when we indigenize technology. So you need to learn to indigenize technology, and that is where universities and scientists are very important.
And also, sir, sorry to take your time. I know you're not feeling well, but very curious to know, especially regarding this CAR T therapy. There's a lot of, I was going through the American Cancer Society website a while ago. There are a lot of side effects, especially with the neurological, as well as it creates the allergies, as well as the mineral imbalance, and the immune system weakens. So, how robust is going to be?
We know, in fact, when we started managing COVID, we know the disease process in COVID is not virus driven. It is the immune system of the human body which goes into overdrive. And that is what results in the cytokine storm. The same cytokine storm occurs when we give CAR T cell therapy to a human being. Okay, so a sort of cytokine release syndrome occurs. And we know that that is what destroys the lung and the blood vessels in the lung in COVID. So we have learned how to avoid that and how to treat it. We have learned that when we used dapsone in leprosy, in lepromatous leprosy, when you use dapsone, you get the same cytokine release syndrome. We know that rituximab used to cause the same syndrome in T-cell lymphomas. Okay, so we know that the same type of immune reaction can occur. It's an overdrive by the body. So we should learn how to control the immune reaction. Just like how we do in a kidney transplant, we are preventing the host from killing the graft. Or in an allograft, we are preventing the transplanted marrow from killing the host. Both are immunological reactions. Same way, we can reduce the intensity of the reaction very successfully if we anticipate and stay one day ahead of the reaction.
Thank you. Thank you. So it is wonderful talk, Dr. Rao. The complete spectrum of therapeutic approaches that are there, the evolution that it has taken place, and what kind of therapies that are going to come in future also you have enlightened us, which we rarely get the opportunity to listen. And see, the most important thing that we always face in India is it is not a lack of intellectual ability of the scientists or the clinicians, but bringing them together is the most difficult part. And government also has systematically isolated them. And no university is having a medical school or clinical lab. And the clinic, wherever the hospitals are there, as you know, you're all, I think, loaded with the patients and no time to interact with the basic scientists. And also basic scientists also, they are very comfortable in their zone, not moving out and making efforts to collaborate and then interact. So when do you think that this kind of system will change?
Sir, the system will change on two fronts, sir. My personal feeling, this is the proposal I have made to Government of India also. Henceforth, the seats into the medical colleges must at least 50% should be reserved for people who have exposure to basic sciences. So I would like master students, like in the West, you can't join MBBS soon after your intermediate. Nobody will take you. There are mathematics branches, humanities branches, there are biological sciences branches, molecular biologists. 80% of the seats go to such people anywhere in the world. So the idea is that this man has an concept of basic sciences and he can use it when he comes to the clinic. So you need to create a platform where the basic scientist can now study medicine and then spend half his time in the lab rather than in the clinic. The second way round is also positive. For example, in Canada, they create what is called a clinician PhD. So, or like in Germany, you are not entitled to put the word doctor after your MBBS, you're only called Mr. So if after your 5 years, if you spend an additional 2 or 3 years working on a PhD, then you can put up the word doctor before your name. So you need to create both ways. You need to create a scientist with a clinical outlook, and you need to create a clinician with a laboratory outlook. Both are equally important, or what we call clinician scientists. So I think this methodology of intake into medicine soon after your 12th standard is bunkum. It doesn't produce anybody except people who learn by rote. They learned by rote in Narayana and Chaitanya. That's exactly what they vomited, and they will continue to do the same thing throughout their lifetime. They are not useful. They are what we call automatons. They learn their medicine from the medical detail man from the pharmaceutical company, not from anywhere else. So we should learn to integrate many different specialties either in the medical college or in the university. Unfortunately, this proposal was there even for the Medical Council of India. It may take some time before it materializes, but I'm sure before that, the clinician and the basic scientist must start collaborating, sir.
Yeah. See, the recruitment of basic scientists in any hospital set up or the medical institutes. See, convincing the MCI is the major problem. No, MCI has so many stringent rules that no basic scientist could be appointed in any of the medical schools or colleges. So convincing MCI is the most difficult kind of thing. Then similarly, Veterinary Council of India is also very difficult to convince them. No basic scientist could be appointed in any of the ICR institutes. So hopefully the government will realize one of the days. So we tried to establish that All India Institute of Medical Sciences within our campus or maybe close to our campus, but we could not succeed in that. Anyway, these are the problems that
There is one
national
question.
level we need to discuss.
So there is one question.
Yeah. So let me congratulate you for a wonderful talk, and we would like to interact. We asked you from Hyderabad, but Vizag is not far off for interaction with any of our basic scientists.
Right.
Thank you.
You're right.
Thank you, sir. It's good to see you.
Thank you.
But there is one question in the message by one of the student, I guess. So what is the efflux rate of drug in antibody-targeted cancer cells?
What is the?
Efflux rate of drug.
You should actually create a drug which definitely deconjugates as soon as it touches the receptor. So I was actually going through the mechanisms of engineering the drug antibody conjugates or the isotope antibody conjugates. So the linker is so important. Majority of the linkers are actually made to disintegrate over a given pH. So you start with the pH of the blood, and as you reach the tumor, the pH is altered. So most of the linkers disintegrate or depolymerize as soon as they come to the acidic pH of the anoxic cancer cell milieu. And that is where it is taken up by the exosome. I did not show you the pH chart and how it is dissociates simply because of time. I have those slides. Actually, I have another 60 slides, but I didn't do that. It will be too boring. But yes, there are mechanisms by which you need to design the linker to be able to separate when it reaches the tumor. You are absolutely right, sir. So it is highly effective, sir.
So are there any questions? So if there are no more questions, I would like to thank you very much, sir, for taking time and giving some knowledge and then bridging the gap as a motto or theme of this World Cancer Day. And as Professor Redana sir said, though it is a loss to Hyderabad, and still we keep continue in collaboration with some of your students actually.
We actually started a collaboration with IIIT, sir. Okay, we are trying to develop some automated systems, including from the basic manufacturing of the instrumentation for cancer screening programs, sir. So although I am at Visakhapatnam, 8 states and 36 centers are collaborating, sir.
Okay. So if there is any basic science part, we will be happy to collaborate.
Very happy, sir.
You
can count me in to help you coordinate that, sir. There is no problem.
Sure. Sure.
Right.
So IIIT is actually within our campus. So
I know,
now
sir.
we are visiting, visitors also in the university.
Definitely, sir. I will. Definitely. Thank you, sir.
Thank you,
So
sir.
we-
It was very enlightening talk. Really
Thank you.
enjoyed it.
Thank you, madam.
And you pumped
Thank
up a
you,
lot
thank
of hope into cancer patients.
Right, madam. Right. Bye-bye, sir.
With that, I would like to conclude by thanking, starts with some of our NSS coordinators, Ms. Kiravani, Mr. Yogesh, Karthik, and others, and for doing all this work, background work. And let me start with our Vice Chancellor, Professor B.J. Rao, and who is the one who made this 30 minutes talk to 2 hours program. And though he was in some other interviews committee, he could make some time and then had given a small talk. And next is Professor Kumar from IISc Bangalore, where he immediately accepted and then spent some time. And then, of course, Dr. Raghunath Rao Garu. And I know he's so busy in the treating the patients and all still. So he was very, I mean, I really appreciated him. He even came forward to help me in the financially if it is required. And as Professor Redana sir was saying, see, we find such a clinicians very rarely because everybody is busy in the clinic and other aspects. So thank you very much, sir, Raghunath sir, and happy to have you here and for enlightening our audience. And Professor Brahmanand, who is also joined with me to organize this. And apart from this, I would like to thank all my colleagues from School of Life Sciences, School of Medical Sciences, and even other schools from the campus, and for joining in this session in awareness of this cancer program. Thank you, Professor Redana Garu, Madam, Sarada Madam, and Prakash Babu, Madhu. There are so many faculty here. Thanks everybody for joining.
Thank you for organizing. Thank you.
Brahmanandam garu. Brahmanandam garu.
Madam.
నిన్నే వచ్చింది నేచర్ లో ఐ సెంట్ ద లింక్ కార్తి maybe you must have seen
Okay,
it.
madam. I'll go through, madam. I will see that.
Must have seen it.
I have not seen this. I'll see now.
Yeah, I just sent the link.
Sure, madam. I will see that. Yeah.
Maybe you should do that onto me. I will volunteer to you.
Okay, madam. Sure. Let me go through this. Yeah.
Yeah, wonderful program, Bindu and Brahmanandam.
Sir, it is actually Bindu organized it, so I just asked you to help.
It's a wonderful talk. Wonderful talk.
Thank you, Ravi.
Thank you, sir.
So
Thank you, sir.
Thank you.
Bye, Bindu. Yeah.
Thank you, Kiravani.
Thank you, Dr. Bindu, very much.
Thank you, madam.
Appreciate it.
Happy to see you. Thank you.